Everyone agrees that fossils are cool. But a few decades ago, a certain franchise introduced us the idea of what if we could extract DNA from these fossils? What could we learn? What could we do? Well, let's talk about what's actually going on with ancient DNA. Ancient DNA, the time is now. So, of course, we're talking about DNA from fossilized organisms. We have a couple things we're going to talk about. First, we're going to talk about what it is. Then, we're going to talk about three specific instances where we've used ancient DNA, namely Neanderthals, Denisovans, and the Cima de los Huesos hominins. But let's get right into it. So, when we're talking about ancient DNA, you might see it abbreviated as simply aDNA, though a lot of people do write it out all the way. Really, we're talking about something that's pretty old. So, there is a fossil, probably a couple thousand years old, that we have now dug up. Because it is so old, there's a couple things going on. First, there's a lot of degradation. First, we're just going to find it in shorter fragments. Like everything after death, molecules begin to break down, and DNA is no different in that instance. We do find DNA, it tends to be best preserved when it is kept cold and dry, so you're more likely to get DNA from fossils in cold regions or that preserved in cold somehow. We also find a couple of diagnostic changes to the base pairs in ancient DNA, and sometimes that actually helps us identify what is ancient and what is modern. And the other thing that happens is there's a lot of contamination. Immediately after death, organisms are colonized by bacteria. So that's why we see a lot of microbial DNA. That's actually not too much of a problem in this instance because their genomes are pretty different from ours. So if we're, say, looking at a fossil early modern human, we can simply identify all the bacteria and just throw it out of our analysis because that's not what we're interested in. We want to look at the genome of that early modern human. What is a little bit more problematic for our purposes is there can be modern human DNA in this sample. So this could be the person who excavated the fossil, or this could be the lab technician. Now that we are seeing more and more uses for ancient DNA, we're actually seeing some people start to excavate in hazmat suits. On one hand, that's really cool, but on the other hand, that sounds so not fun. Excavation is already really hard, and now we're adding the complication of if you have to do that in the big, ungangly hazmat suit. I'm glad that's not my job, but thank you, people who do that for us geneticists. Thankfully, at least on the lab side, we already have clean room procedures that are well established. So, in all labs that process a lot of ancient DNA, they'll have a clean room to help prevent as much contamination as possible. Um, In this lecture and this class, we're mostly concerned with true ancient DNA, so something extracted from a fossil, but we actually use similar DNA extraction methods even when we're just using museum specimens that may be only, a few hundred to even a few decades old. So here are some museum specimens I extracted. These are dried skin samples. And one of the interesting things is we have a couple extra steps when we are extracting from these older specimens. Um, you can see it here. I'm pouring liquid nitrogen on top of our samples, and this helps freeze it, and yeah, it makes it better. Well, this is one of the weird things about science. Sometimes there's like myths around lab work. This is just something we do. I've never been able to find a clear explanation as to why, but yeah, we freeze it. But makes me feel like a little bit of a mad scientist here. Um, the oldest hominin that we've been able to extract DNA from is 300,000 years old. That's the Cima de los Huesos material. Though the oldest ancient DNA we have is 700,000 years ago, and this is a horse ancestor. But we, of course, have ancient DNA from so many different things at this point. The first ones were experiments on Egyptian mummies, trying to, perfect our methods. But we also have early modern humans, Neanderthals, Denisovans, Cima de los Huesos hominins, and we have a bunch of non-human stuff. We have cave bears, we have giant moa from New Zealand, which are now extinct. We have dog ancestors, we have horse ancestors, and probably other ones that I don't even know about yet, because that's just how much ancient DNA there is. Since this methodology came out in the past few decades, like it has exploded, and it is a burgeoning field in and of itself. So at this point, you might be wondering, can Jurassic Park happen? The sad part is probably not. First, DNA actually doesn't preserve very well in amber. So, first, talking about the specific way that happened in Jurassic Park, taking blood from a mosquito preserved in amber, it's probably not going to survive. This was actually published earlier this year, though, that someone was able to extract DNA from an organism embedded in resin, but that was only 6 years old. There's a little bit of a difference. And also remember, the dinosaurs went extinct 65 million years ago, so that means all the dinosaurs are older than 65 million years ago. And the oldest ancient DNA we have is only 700,000 years old. So that's, a couple orders of magnitude in difference there. I just don't think we're ever going to be able to get DNA from a fossil dinosaur. There are, of course, other ways to make dinosaurs. Check up Evo Devo on making chickens with teeth, but that's a different field. Let's get back to ancient DNA though. Like, why are we doing this in the first place? If we can't make dinosaurs, like, really, what's the point? What we're trying to do here is now it gives us the opportunity to compare genetic lineages at different points in time. So it can be like, are they the same? Are they related? It actually makes it possible to sample the potential ancestors of modern species. And we can also sample lineages that are now extinct. So we can get information that we would never be able to see if we were only looking at modern species. When we're talking about this, it is also important to consider that there are different types of DNA. In all of our cells or eukaryotic cells, there are two types. We have nuclear DNA. That's generally what is considered your genome, but the mitochondria, that powerhouse in your cell, has its own genome. So we actually have two genomes. And because there are multiple mitochondria in your cell, there's a lot more mitochondrial DNA in you and in every cell. They're also inherited differently because you get the cytoplasm and pretty much all of the cellular material from your mother, you get the mitochondrial genome from your mother as well. So nuclear DNA, of course, you get from all of your ancestors because you get half from your dad, half from your mom, they combine, boom, it's you. But again, you only get mitochondrial DNA from your mother. So that is the maternal lineage. And that means we can kind of compare these two different lineages and look at how they are different. And that can give us some insights into some really cool things. Also remember that your nuclear genome recombines. So this is the process of crossing over that happens during meiosis. And that means if you look back a couple generations that we are really a composite of our ancestors. And through recombination processes like this, the farther back you go, you're actually less likely to have DNA from any one specific ancestor because you could simply lose chunks of DNA from that ancestor due to recombination. But let's summarize some of our differences here between mitochondrial and nuclear DNA. Remember, there's a lot of mitochondrial DNA in every cell because we have a lot of mitochondria. And there's only one nuclear genome per cell. Mitochondrial DNA is maternally inherited. Nuclear DNA you get from both of your parents, and we have recombination. Because there is no recombination in our mitochondria, that's actually just a single locus. But in our nuclear DNA, we have many loci, so we can compare them and see what's going on between them. Mitochondrial DNA also has a very high mutation rate, whereas the nuclear genome has a much lower mutation rate. So this allows us to say slightly different things. All in all, this means mitochondrial DNA is very easy to extract. So if you're trying something for the first time, you're probably going to go for the mitochondrial DNA first. And actually, most of the early stuff on ancient DNA was mitochondrial. But because of that, single locus, high mutation rate, we can pretty much only say much about recent ancestors. Whereas for the nuclear DNA, it is more difficult to extract, but there's so much more data there. We're able to look farther back in time. We're able to compare different loci. So really, we do want to get both, and the nuclear DNA is kind of that gold standard. That's the thing we really want to get at. If you want to talk about ancient DNA and how it's impacted our understanding of human evolution, there are two papers you want to read. First, this summary paper by Todd Disotell in 2012. Of course, it's 8 years old now, so it, of course, doesn't cover the 8 years after this was published, but this is a great first step to understand some of the basic methodology and the early stuff. Now, there's also a more recent paper from 2020 that kind of summarizes all of this and what we know today. So I highly recommend you check out both these papers to get a better idea of what ancient DNA is and how it's impacted our understanding of human evolution. So, can you explain what is ancient DNA and what can we learn from it?